Welcome back. We are at the last chapter, we are at the last lecture of chapter number 4, where we are discussing fundamentals of biospectroscopy and bioimaging. And with this lecture, I will be concluding this particular chapter, and thereby in this particular lecture, I will be giving you primary examples of different type of biospectroscopy and bioimaging. Specifically, I have taken two because I personally use them too, not that they are the best, but I use them. The results that are shown, several of the results, not all, are mine own, and therefore I can probably teach this thing slightly better. So, the first one is optical coherence tomography. You might have seen or heard of optical coherence tomography happening when you visit an ophthalmologist. Look how simple the overall mechanism is of optical coherence tomography. Remember, coherence is the ability, it's a fixed relationship between two different wavelengths. Either they maintain same phase with respect to one another as they travel, or they maintain the same amplitude as they travel through distance with respect to one another. So, what happens is that in optical coherence tomography, OCT, you have a broadband light source, okay, a light source. At a time you can give one wavelength, or you can have bunch of wavelength, that hardly matters at this present moment, but say for simplicity's sake, you are sending one wavelength of light, visible red light or visible green light or something infrared or visible, that is passed through an optical beam splitter. So, the same wave divided into two part, One is passed through a reference arm into a mirror, a movable mirror, and the another one, which is a sample arm, that is made to pass through a biological tissue, a biological matter, right. It could be anything, it could be tissue, it could be cell, it could be some part of your body, for example, eye or nail or anything. Consider your eye, your eyes, you put your eye in here, right. Now, there are mirrors, of course, mirrors associated with it. So, there will be a back reflection. This is an axial mirror, this is a mirror, this symbol is that of mirror. So, light will pass through the reference arm, puts inside the mirror and will return back. These are fiber, these rays are fiber, optical fiber, which sends light. Similarly, in the case of this mirror and this, the light will be reflected back from the biological matter reflects, scatters, etc. And it is then again recollected at the beam splitter. So, light comes, it divides into two parts, one hits the mirror, one hits the biological tissue, reflects back and recollected at the optical mirror. Now, in order to have a particular coherent relationship, the light that is hitting this and returning back, as well as light this is hitting the biological matter and coming back, need to be fixed in order to have a coherence relationship. The distance travelled by this and the distance travelled by this has to be same for a coherence, for a proper positive interference, it could be negative as well, positive interference to happen. A coherence relationship will only maintain when the distance travelled by this is equivalent to the distance travelled by this. You do not know how deep it is, this biological matter, or how different types of layers are there, or what wavelengths penetrates, what wavelength does not penetrate, but this is movable. At one particular wavelength of light, this has returned back. You are now adjusting it like the screw, like the knob, you are adjusting it like that, so that the light coming from here also matches the light here. From a reference point, you have moved it to few centimeters and you have understood that this is matching. Another wavelength of light, you again readjust it. So, by adjusting this part, you will know its counterpart what exactly is its thickness, what exactly is its layered formation, what exactly how viscous or how many layers or what exactly are the refractive index, etc. All of those things of the sample can simply be generated. So, coherence, a fixed relationship between the phase of wave in a beam of radiation of single frequency. As I said, single frequency, one light at a time. Two beams of lights are coherent when the phase difference between the wave is constant. They are non-coherent if they are randomly changing. Stable interference patterns are formed only by radiation emitted by coherent sources, ordinarily produced by splitting a single beam into two or more beams. So, by adjusting this, because coherence will only happen when the distance travelled is equal to the distance travelled here. So, by changing this, you can understand this, because this is equivalent to this. I will give you an example or advantages. They are pretty high resolution, they have very real time imaging, endoscope, etc. All of these things are done, but the imaging is this, right. This is the optical coherence tomograph of a fingertip. You have put a finger past different types of light and adjusted that, and you have figured out one layer at a time and have reconstructed the entire thing using your computer. These spiral things that are moving, the spiral, those are glands inside your fingertip. It's flesh, blood. I think the bone is missing, it will go little bit below because they have probably seen this part, and then there are small glands, sweat glands, etc., and they have measured it. This is the ocular retinal thickness of an eye. You need to understand, usually OCT is very common in case of ophthalmologist, as I was saying, that you sit in front of a computer, you sit in front of an OCT machine that sends light, tune it in this direction. Now they are all computer controlled, you do not have to manually tune it anything, and it figures out your thickness or the penetration depth of your eye till retina and try to see if there are any obstacle in between, or if there is some formation, cataract or anything, though cataract can be easily seen just by looking into it. But I mean initial stages, if there is a standard distance, standard image of a normal eye or a normal fingertip or a normal retina, if it changes as detected by OCT, then you try to understand, you try to prescribe medicine, you try to do some intervention, therapeutic intervention towards its early detection of glaucoma, etc. All could be done using OCT. The final one where spectroscopy and imaging are combined. So, thus far understand this. This is example number 2. We have discussed spectroscopy, we have discussed microscopy. If you combine them together, you get microspectroscopy. Microspectroscopy is something that I am dealing with regularly in order to do something which is called clinical spectroscopy or clinical diagnostics. Microspectroscopy utilizes the best of both worlds. It uses the imaging resolution from microscope as well as the spectroscopic information from a spectrometer, and thereby we create this microspectroscope. This is my student Shobhik Das. He is also the TA of this particular course. He is very single, so anyone, if you want, contact him directly. So, things you have to do as a supervisor. So, anyways, I told you molecules absorb light and they vibrate. These vibrations are very fixed and they can be measured wave number with respect to absorbance intensity. So, that can be done using a spectrometer, but now we have microspectrometer. I have discussed about FTIR. In microspectrometer, what you can do, you can focus your microscope at a specific area of the sample, illuminate the light, illuminate that area with light, and that reflected light is then collected, understood what kind of lights are missing, what kind of light had been absorbed in reflection mode. In reflection mode, transmission is 0. So, 1 is equal to A plus T plus R. If T is equal to 0, then 1 is equal to A plus R. You have 1, you have R, 1 minus R is A. You figure it out. A is these lambda 1, lambda 2, these are the specific frequency of molecules, you understand the molecule. With a microscope, the advantage here is that you are able to focus at a specific point of a big sample, say a tissue, I will show you, and find out the chemistry from that illuminated point only. Then you go to the next point, then you go to the next point, so on and so forth. So, that is what is called clinical spectroscopy. Primarily what we do previously, what physicians used to do, they take a biopsy, the biopsy from a skin or liver or an organ, then they stain it, yeah, and based on this staining you try to understand if it is a normal, if it is abnormal, if there is a disease, not disease, and this is very, very subjective depending on your pathologist, depending on what place you have been to, what institute you have been to, etc., and then the treatment is done. Whereas, in case of microspectroscope, what you do is you point your microscope, this is a tissue, this is an oral tissue, mouth tissue. We were trying to detect, these are our results, we were trying to detect cancer. So, you focus on this point, then this point, then this point, then this point, you figure out chemistry of this area, you figure out chemistry of this area, you figure out chemistry of this area, you will get roughly graphs like this. Each peak represents proteins, lipids, nucleic acids. You can assign a particular false color to any of those peaks and try to see if this peak is present here, then it is red. If this peak is present here, no, it is not red, and so on and so forth, and thereby you map the entire area. It is a level free biochemical image to target a particular area. Very, very clever and very, very easy. We thereby can create a 3-dimensional hypercube, so called hyperspectral microscopy or hyperspectral imaging, in which each point you can illuminate with one wavelength or several wavelengths. Say for example, a particular wavelength is absorbed by amide, absorbed by proteins only. You have used that particular light to illuminate all these area. It will show the distribution of protein. This area has high amount of proteins, so high intensity. This amount of low amount of protein, low intensity. Then you illuminate it with another wavelength, which is the wavelength absorbed by fat, lipids. This area high intensity, very high fat, this area low intensity, very low fat, and thereby you map pixel wise the entire area, and you can differentiate the entire thing between proteins, nucleic acids and lipids and get a full 3-dimensional chemical image. A chemical image gives you chemical information. Previously you are getting physical information, how big it is, how long it is, depending on the contrast, depending on the size, shape, size, dimension, structures, and you were coming to a medical information, medical diagnostics based on the physical part of it. Okay, the cell has elongated, the cell has contracted, so maybe this because of this disease. Now we are saying that the amount of a particular protein in that particular cell has reduced or increased. So you are giving chemical information. Fat has increased tremendously in this area. Instead of saying that the cell has swollen up or the cell is looking abnormal, you can say these areas, there are lipid formation. White means very, very high, blue means very, very low, white showing that huge amount of lipid formation has happened in this particular area. Remember, especially when we talk about diseases such as cancers, it spreads heterogeneously. Only at last stage everything is same, but at the very beginning this area is cancerous, this area is not, especially in case of oral cancer. So this is a beautiful image that we are about to publish in our work, normal versus oral squamous carcinoma. This is precancerous cell. These are the microscope image. Tell me something, Would any one of you, just by looking at these two images, be able to say just by yourself which one is cancerous and which one is normal? You require training, you require patience, you require degree, etc., all of that to do that, and still you can do mistakes if they are very close to one another. However, we have differentiated into different forms of protein. Protein can have alpha helical form or beta sheet type of form. Same protein can modify its it can fold or it can misfold into different arrangements. One is alpha and one is beta. So our hypothesis is beta is not necessarily good. More amount of alpha could be better, but here, just by looking at the microscope image, you will not be able to detect a biomarker or the protein folding chemistry or anything like that. But using a chemical image, using a perfect chemical image, you will be able to see which one is, say, red is bad. If I say, you can differentiate between these two, or which area is better, which area is not, and thereby you can diagnose it. So this is very latest, very new, our own work, which describes microspectroscopy. Previously we were doing it two separately, microscopy separately and spectroscopy separately. Now we have combined them, and the result is this. If I simply as a layman tell you that if you find red color, it is bad, more amount of red is more bad, you can simply, anybody can say which one is better, which one is not better, and then you apply machine learning, AI, ML, etc. to it. So human intervention is not required. You simply take a slide, put it under this microspectroscope, image it, output is here, and thereby you can intervene. You can say which one is working at what particular condition you have found this the pathological slides as. So I will try to finish it here. I think I had given you enough information about spectroscopy as well as imaging. So these are my references. Previous works were all mine, our team, Nanobiophotonics at IIT Kharagpur, and I'll see you in the next class. Thank you very much.